Welcome back to the deep dive. Today, we're going to explore two bacterial species. They kind of fly under the radar sometimes, but they can be tricky in the clinic, especially for kids. We're talking Moraxella catarrhalis and Kingella kingae, and we're using Gillespie and Hawkey's principles and practice of clinical bacteriology as our guide.
It's interesting how these organisms, they used to be thought of as commensals, mostly harmless, but more and more we're seeing they can cause some real problems.
Yeah, and M. catarrhalis, it's had this really crazy taxonomic journey. It's like it couldn't make up its mind, bouncing between Neisseria, Brennhamella, before finally landing on Moraxella.
Right, and that kind of, that mirrors how our understanding of it has changed too. At first it was just a harmless bystander. Now we know it can cause infections, especially respiratory ones.
Definitely. Keeps us on our toes. And then there's Kingella kingae, part of that HACE group, causing those infections that are so hard to diagnose.
Yeah, kingae. It had a name change too. It was Moraxella kingae at first. It's a pretty picky bug in the lab. Needs just the right conditions to grow. That can make it easy to misidentify it. I've even seen it mistaken for Neisseria gonorrhea a couple times.
Oh, wow. That's a bit scary to think about. Lab mix-ups like that. Okay, so we've got these two bacteria, one with an identity crisis, the other a picky eater. What about their ability to cause disease, their pathogenicity?
Well, for a while there, it was hard to even study that properly. We didn't have good ways to type them. The early methods like serotyping or using outer membrane protein profiles, those had a lot of problems. But then things changed. We got these molecular typing methods, REA, PFGE, AFLP, automated ribotyping, all based on nucleic acid polymorphisms. Those really changed the game.
So we finally have the tools to really track them, huh? What did those methods show us? Like, what did we learn about these bacteria?
With M. catarrhalis, we saw there's a lot of genetic diversity, different clones circulating in different populations, different places. That's huge for understanding how outbreaks happen. And it showed us how horizontal gene transfer plays a role in its evolution. That's probably part of why it can become resistant to antibiotics so quickly.
Wow, that's really important for anyone working in the lab to know. What about K. kingae? Did those molecular methods help us understand it better too?
Yeah, maybe not as much as with M. catarrhalis, but PFGE and ribotyping, those have been helpful, especially for tracking outbreaks, like in daycare centers.
Daycare centers, right. That brings us to something I was surprised by. M. catarrhalis, it's super common in kids, but not so much in adults. Why is that? Why the age difference?
The immune system, probably. Kids, their secretory IgA antibodies, those are key for mucosal immunity. They aren't fully developed yet. As they get older, those antibodies mature and the carriage rate goes down. Plus, there's a seasonal thing. More carriage in winter and autumn, maybe because of changes in immunity or just being indoors, more, closer contact.
So basically, kids are like little M. catarrhalis carriers, especially when school starts back up. What about K. kingae? Does it follow the same pattern?
It's similar, yeah. Like H. influenza, K. kingae carriage is highest in kids under 3, especially in daycare. And like you said about M. catarrhalis, secretory IgA. As that develops, carriage goes down. But there's this weird thing. Carriage rates are similar in boys and girls, but boys get infected way more often. Not really sure why that is.
Huh. That is weird. Hope someone figures that out. Let's talk about the actual diseases these bacteria cause, the clinical features. What do we see in kids?
Catarrhalis, it's a big player in those childhood respiratory infections, especially sinusitis and otitis media. It's actually the most common bacterial cause of middle ear infections in kids.
Right. I was reading that M. catarrhalis is tricky. Sometimes PCR will find its DNA in the middle ear fluid, but the culture is negative.
Yeah, that's a good point. Suggests it might be there causing the infection, even when we can't grow it easily with our usual methods. And it's not just ear infections, it can cause lower respiratory tract infections too. Bronchitis, pneumonia, especially in kids with other health problems.
Definitely not something to ignore then. What about K. kingae? How does it show up in kids?
K. kingae is, it can cause a whole range of things in kids. Bone and joint infections, endocarditis, bacteremia, even meningitis, though that's less common. The thing is, it seems to really like bones, joints, and the heart. It has this tropism for those osteoarticular and cardiac sites.
So some serious stuff. We've talked about kids. What about adults? Do these bacteria still cause problems as we age?
They do, yeah. Not as common as in kids, but M. catarrhalis can cause problems in adults with COPD, making it worse. And it can cause laryngitis, bronchitis, pneumonia. It's even been found in nosocomial infections.
Hospital-acquired, yikes. That's a worry, especially for people with weaker immune systems. And K. kingae in adults.
In adults, it's mostly bacteremia and endocarditis. Those can be really serious if you don't catch them. And the tricky thing is, Kingella infections, they can look really different from person to person, making it hard to diagnose and treat.
Seems like we got to watch out for these bacteria no matter the age. All right, let's talk lab diagnosis. How do we pin down these tricky bugs?
Catarrhalis, it can be tough to isolate it, especially from sputum samples. You've got all these other Neisseria species hanging around that aren't pathogenic, so we often have to use a special kind of agar, a selective media.
Selective media, what's the secret ingredient there?
Well, it has things like acetazolamide, vancomycin, trimethoprim, and amphotericin B. They stop those other Neisseria from growing so the M. catarrhalis can stand out.
So it's like a microbial bouncer. Keeps the riffraff out. So once we maybe have M. catarrhalis on this special plate, what then? How do we know for sure it's the right one?
Then we go back to our basic microbiology techniques. Gram staining first to get some visual clues. M. catarrhalis usually looks like gram-negative diplocacci, often with kind of flattened sides, but sometimes it holds on to the crystal violet stain. Tricky little bugger.
Likes to keep us guessing. What else do you do?
We look at the colonies, how they grow, and of course, biochemical tests, oxidase production, DNase production, whether it can ferment different sugars, all those classic tests. When those line up, we're pretty confident it's M. catarrhalis.
Real detective work. What about K. kingae? Is it as easy to ID?
Not so much. Remember, this one's picky. Regular blood agar, it often won't grow on that.
So how do we get it to grow? What does it want?
We often use those Bactac blood culture bottles that have the extra nutrients it needs, and even then, it can be slow. Really test your patience.
You got to cater to its every whim, huh? Anything else we can use to ID K. kingae?
PCR can be really helpful, just like with M. catarrhalis, especially for joint fluid or blood samples. It's much more sensitive than culture. Can pick up cases where there's just a tiny amount of bacteria, too little to grow in culture.
So PCR, it's like our molecular magnifying glass. Okay, we've talked about identifying them. What about how they spread, the epidemiology? Who's at risk for these infections?
Well, M. catarrhalis, it spreads through respiratory droplets. That means being close together, like in a daycare or at home. That really increases the risk.
Right, so sharing toys, close quarters, that's prime territory for M. catarrhalis. Sounds like kids are spreading it to each other, and maybe to adults too, especially if the adults have weak immune systems or lung problems. How about K. kingae? Same kind of spread.
Yeah, respiratory droplets, same idea. Daycare is a big place for transmission. All those kids close together, it really helps the bacteria spread.
Makes sense. Keeping hands clean, covering coughs, that's got to be key to stopping it. What about prevention? Anything else we can do?
Hygiene is really the most important thing. Hand washing, covering coughs and sneezes, cleaning surfaces, all that basic stuff, it really does help reduce transmission.
Good old-fashioned hygiene. Still the best. What about vaccines? Any shots for M. catarrhalis or K. kingae?
Not yet, unfortunately. No licensed vaccines for either one. But there's research going on, some promising candidates. Scientists are working on vaccines that target those key virulence factors, the outer membrane proteins, lipoglycosaccharides, trying to create something that will protect against infection.
Always good to hear about vaccine research. Fingers crossed. All right, let's talk treatment. What's the usual approach for these infections?
With M. catarrhalis, antibiotic resistance is a big problem. Used to be, it was easy to treat with penicillin. But now, most strains produce beta-lactamases. Those enzymes break down penicillin, make it useless.
It's like they're evolving right in front of us, so what do?
We have to use different antibiotics. Amoxicillin clavulin often works, or we use a beta-lactamase inhibitor along with penicillin. It's a constant battle, trying to stay ahead of their resistance.
A real microbial arms race. What about K. kingae? As resistant as M. catarrhalis?
Luckily, no. K. kingae is usually more sensitive to antibiotics. Penicillin, ampicillin, those often work. But there is some resistance out there to older quinolones, trimethoprim, sometimes even vancomycin.
So we can't let our guard down completely.
Definitely not. We always have to test for susceptibility and make sure we're using the right antibiotic.
Got to be strategic. Okay, beyond antibiotics, what else is important for managing these infections?
Supportive care is important. That means pain relief, fever reducers, making sure the patient stays hydrated. All that basic stuff makes a big difference.
The simple things. Anything specific we should do depending on what kind of infection it is?
Oh, for sure. Respiratory infections, rest, and fluids are really important. For more serious things, like those bone and joint infections K. kingae can cause, sometimes you need surgery. Draining an abscess or removing infected tissue.
So it's not just about killing the bacteria. It's about helping the body heal, too. We've learned a ton today, from their weird taxonomy to how we diagnose them, treat them, and deal with that pesky resistance. M. catarrhalis and K. kingae, they might be small, but they can cause some serious trouble.
They're definitely not organisms to underestimate.
All right, let's test our listeners' knowledge. Ready for a pop quiz?
Always ready. Let's see what they've learned.
Okay, first question. What's the main way Moraxella catarrhalis becomes resistant to penicillin? Think back to those enzymes we talked about. Take a moment to think.
This is a big one. It's important to understand how this resistance works.
Time's up. The answer is it produces beta-lactamases. Remember, those enzymes break down penicillin, make it ineffective. That's why we have to use other antibiotics or add a beta-lactamase inhibitor to the penicillin.
Exactly. You got to know your enemy. Okay, question 2. Which age group is most likely to carry Kingella kingae? Think back to our discussion about daycare.
This is a key fact, especially if you're working with kids.
Absolutely. Knowing who's at risk is important.
Time's up. The answer is children aged 0-3 years, especially those in daycare. That close contact, it really helps the bacteria spread.
Right. Makes those early years a high-risk time. Okay, question 3. This one's a little tougher. What's one of the main ways Moraxella catarrhalis evades our immune system? Think about how our body fights off bacteria.
This is about how sneaky bacteria can be. They've got all these tricks.
It's a constant arms race between our immune system and these microbes.
All right, the answer is complement resistance. Our complement system is a powerful defense mechanism. But M. catarrhalis has found ways to resist it, to survive in our bodies.
It's a wily one for sure. Multiple ways it can do this too.
Okay, question 4. This one's about diagnostics. What's the most sensitive test for finding Moraxella catarrhalis in middle ear fluid? Think about those cases where the usual methods might not work.
Sometimes you need that extra sensitive tool to get the right answer.
And the answer is PCR. It can detect tiny amounts of M. catarrhalis DNA even when we can't grow it in culture.
PCR is a lifesaver in those situations. Gives us that extra level of confidence.
Last question. This one's about K. kingae and its habits. Which 2 parts of the body does Kingella kingae tend to infect? Remember, it seems to have a preference for certain areas.
Knowing this pattern can help us diagnose it faster and start the right treatments sooner.
Time's up. The answer is osteoarticular and cardiac sites, bones, joints, and the heart. K. kingae really seems to like those spots.
Exactly. Keep that in mind, especially if you're seeing a patient with symptoms in those areas.
All right, that's our quiz. Hopefully that was a fun way to test your knowledge.
Good way to solidify what we've talked about today.
It's amazing we've covered so much about M. catarrhalis and K. kingae, from their confusing taxonomy to their sneaky virulence tricks and how their resistance keeps evolving. It really shows you can't underestimate these bacteria.
Definitely not. Always learning, always adapting.
That's the life of a microbiologist.
It makes you wonder what we'll discover next. What other little secrets are hiding in the microbial world?
Right. Always something new to learn. So speaking of uncovering those secrets, let's talk about the lab side of things. Diagnosing these 2 bacteria, I was reading that it can be a bit tricky.
Oh yeah, for sure. Isolating M. catarrhalis, that can be a real pain, especially from sputum. It loves to hide amongst all those non-pathogenic Neisseria species.
Like a needle in a haystack.
Yeah.
So how do we find it?
We have to get a little creative with our media. Use what we call selective media. It's basically like a special agar that only lets the bacteria we want grow.
So it's picky. The agar itself is picky. What's in it that makes it so special?
Well, we add things like acetazolamide, vancomycin, trimethoprim, amphotericin B. Those inhibit the growth of those other Neisseria species. Gives M. catarrhalis a chance to take center stage.
So it's like a velvet rope for microbes. Only the VIPs get in.
Yeah.
Okay, so let's say we've got something growing on our selective media. How do we know for sure it's M. catarrhalis?
Then we put on our detective hats and go back to the basics. Gram staining, that's always our first step. Gives us those initial clues. M. catarrhalis is usually a gram-negative diplococcus, often with those kind of flattened sides. But it can be a bit of a trickster sometimes, occasionally holds on to the crystal violet stain.
A bit of a rebel, huh? What other tests do we have in our arsenal?
We also look at the colony morphology, how it grows on the plate, and of course the biochemical tests, the classics, oxidase production, DNase, sugar fermentation patterns, all those help us put the pieces together. When everything lines up, then we can be pretty confident we've got M. catarrhalis.
It's like solving a puzzle, one test at a time. And what about our picky eater, K. kingae? Is it as straightforward?
Not quite as easygoing.
Okay.
K. kingae, it's very particular, regular blood agar, often won't even grow on it.
High maintenance bug. What do we do then? What does it need to thrive?
We often have to use those Bactec blood culture bottles. They have the extra nutrients it seems to like. Even then, it can take its sweet time growing. Really test your patience.
Spoiled little bug. Anything else that can help us IDK kingae?
PCR can be incredibly helpful here, just like with M. catarrhalis, especially when we're working with joint fluid or blood samples. PCR is it's so much more sensitive than culture, can pick up even the tiniest amounts of bacteria.
Our microbial detective dogs sniffing out those hidden traces. Okay, we've talked about IDing them, but what about the bigger picture, the epidemiology? How do these bacteria spread and who's most at risk?
Well, M. catarrhalis, it's a bit sneaky. It mainly spreads through those tiny respiratory droplets. So being close together, like in a daycare or a household, that really increases the chances of transmission.
Sharing toys, coughing on each other, all those lovely kid things. Sounds like kids are the main spreaders then, passing around to each other and maybe to adults with weaker immune systems or lung problems. What about K. kingae? Same story.
Pretty much, yeah. Respiratory droplets, close contact, the whole 9 yards. Daycare is a prime spot for transmission. All those little ones in close quarters.
Breeding ground for bugs. So keeping things clean, covering those coughs, those are got to be crucial. What about prevention in general? Anything else we can do?
Good hygiene. That's really the cornerstone. Hand washing, covering coughs and sneezes, keeping surfaces clean. All those basic things really do make a difference.
Simple but effective.
Yeah.
What about vaccines? Any hope on that front for M. catarrhalis or K. kingae? I know a lot of parents would be thrilled.
Unfortunately, not yet. No licensed vaccines for either one at the moment. But there's a lot of research going on, some promising candidates in the pipeline. Scientists they're really focusing on those key virulence factors, the outer membrane proteins, lipoligosaccharides, trying to develop something that will provide real protection.
Fingers crossed they find something soon. Those vaccines would be a game changer for sure. All right, let's talk treatment. What's the usual approach for these infections?
With M. catarrhalis, we have to be really careful because of antibiotic resistance. It used to be so easy to treat, just a course of penicillin, good as new. But now it's most strains produce those pesky beta-lactamases.
Those enzymes that break down penicillin, right? So what do we do now?
We have to use other antibiotics. Amoxicillin clavulinate, that's often a good choice. Or we can use a beta-lactamase inhibitor along with penicillin. It's a constant battle trying to stay one step ahead of those evolving bacteria.
The microbial arms race. What about K. kingae? Is it as resistant as M. catarrhalis?
Thankfully, no. K. kingae is usually much more sensitive to antibiotics. Penicillin, ampicillin, those often work well. But there is some resistance out there to older quinolones, trimethoprim, and even vancomycin in some cases.
So we can't get complacent. Always got to be testing.
Absolutely. Susceptibility testing is key. Always want to make sure we're using the right antibiotic for the job.
Smart move.
Oh.
Okay, beyond the antibiotics, what else do we need to think about when managing these infections?
Supportive care, that's always important. Making sure the patient is comfortable, managing pain and fever, keeping them hydrated. Those basic things make a world of difference.
Back to basics. Sometimes those are the most important things. Anything specific we need to do depending on the type of infection?
Oh, yeah, for sure. For respiratory infections, rest and fluids, those are key for recovery. But for those more serious infections, like the bone and joint infections K. kingae can cause, sometimes surgery is necessary. Draining an abscess or removing infected tissue, that can be really important to prevent further spread.
So sometimes it's not just about the bugs themselves. It's about the damage they've done, the complications. Well, we've covered so much ground today, from taxonomy to diagnostics, treatment, and management. M. catarrhalis and K. kingae, they might be tiny, but they can certainly pack a punch.
They're a good reminder that we can't let our guard down. The microbial world is always full of surprises.
Always keeps us on our toes. Before we wrap up, let's have a little fun. Time for a pop quiz. Ready to test our listeners' knowledge.
Always up for a challenge. Let's see if they've been paying attention.
All right, question number 1. What's the main mechanism of resistance to penicillin in Moraxella catarrhalis? Think back to those pesky enzymes we discussed earlier. Give everyone a moment to think.
This is a big one. Understanding how that resistance works is crucial.
Time's up. The answer is beta-lactamase production. Remember those enzymes that break down penicillin and make it useless? Well, M. catarrhalis has become quite adept at producing them. That's why we often have to use other antibiotics or pair penicillin with the beta-lactamase inhibitor to overcome that resistance.
Exactly. Got to know your enemy. Right. All right. Question number 2. Which age group has the highest carriage rate of Kingella, the kingae? Think back to our discussion about those close contact environments. This one's especially important for anyone working with pediatric patients.
Knowing who's at highest risk can really help guide our approach.
Absolutely. It's all about understanding those patterns.
All right, time's up. The answer is children aged 0–3 years, especially those in daycare settings. That close contact really facilitates the spread of kingae.
It's a good reminder that even though kingae might not be as common as some other pathogens, it can still pose a significant threat to this vulnerable age group.
Ready for question number 3? This one delves a bit deeper into the world of bacterial virulence. What's a key virulence factor of Moraxella catarrhalis that helps it evade those powerful host immune defenses? Think back to our discussion about how bacteria can be so sneaky and resilient.
Bacteria have evolved some incredible strategies to survive within our bodies. This question really highlights that.
It's a constant battle between our immune system and those crafty microbes. All right, the answer is complement resistance. Our complement system is a vital part of our immune response. It's a complex cascade of proteins that can target and destroy invaders. But M. catarrhalis has found ways to resist this system, allowing it to persist within the host.
It's quite a clever little bugger. And it can do this in multiple ways, which makes it even trickier to deal with.
All right, on to question number 4. This one focuses on the crucial role of diagnostics. Which diagnostic method is considered the most sensitive for detecting Moraxella catarrhalis in middle ear effusions? Remember our discussion about the limitations of traditional culture methods.
Choosing the right diagnostic tool is crucial for accurate and timely diagnosis. Sometimes those traditional methods just don't cut it.
We need those specialized tools to give us that extra edge. And the answer is PCR. PCR can detect even tiny amounts of M. catarrhalis DNA, even in cases where culture methods fail to grow the bacteria. This makes it incredibly valuable for diagnosing middle ear infections, especially when traditional cultures come back negative.
PCR is a true game changer in the clinical microbiology lab. It gives us that extra level of sensitivity and confidence we need to make the right call.
All right, ready for our final challenge? Question number 5 is all about kingae, and its, let's call it, predictability. Kingae is known to have a particular tropism for which 2 anatomical sites. Remember, tropism refers to a microbe's tendency to preferentially infect certain parts of the body.
Recognizing these patterns can be incredibly helpful in guiding our diagnostic thinking and treatment decisions. It's like having a roadmap of sorts.
It definitely helps us narrow down the possibilities and target our approach. Time's up. The answer is osteoarticular and cardiac sites, bones, joints, and the heart. Kingae seems to have a special affinity for these areas.
Exactly. This is crucial information for clinicians to keep in mind, especially when they're seeing patients with symptoms that could be related to these anatomical sites. Early recognition and intervention can make a big difference.
Well, that wraps up our quiz for today. Hopefully our listeners found those questions both challenging and informative.
It's a great way to test your knowledge and reinforce those key concepts.
It's amazing how much ground we've covered in this deep dive. From those early taxonomic mysteries to the intricacies of lab diagnosis and treatment and the ever-present challenge of antibiotic resistance. M. catarrhalis and K. kingae, they might be small, but they're certainly not to be underestimated.
They're a constant reminder that we have to stay vigilant and adaptable in our field. The microbial world is always changing, and we need to be ready to evolve with it.
On that note of evolution and adaptation, I think it's time to wrap up our deep dive into the world of Moraxella catarrhalis and Kingella kingae.
It's been a fascinating journey exploring the hidden complexities of these 2 intriguing bacteria.
A big thank you to all our listeners for joining us on this adventure of microbial discovery. Until next time, stay curious, keep those labs buzzing, and those microscopes focused. We'll catch you on the next deep dive. It really is remarkable, these tiny, tiny creatures. We can't even see them without a microscope, but they can cause so much trouble.
It's a humbling reminder, isn't it, of just how complex and powerful the microbial world is and how crucial our role is as medical microbiologists in understanding and combating these organisms.
Absolutely. Today's deep dive, it's been a perfect example of that. We've explored the ins and outs of Moraxella catarrhalis and Kingella kingae, learned about their quirks, their potential to cause disease, and the challenges we face in diagnosing and treating those infections.
Oh, we've gone from their their taxonomic journeys, those name changes and reclassifications, to their sneaky ways of evading our immune systems, and the constant battle against antibiotic resistance. It's a lot to take in, but it's essential knowledge for anyone working in this field.
Even for someone like me who talks about microbes all the time, I got to say, I've learned a lot today. Really makes you appreciate just how much there still is to discover about these tiny but mighty creatures.
That's what keeps it exciting, right? The microbial world is always changing, always evolving. There's always a new puzzle to solve, a new challenge to overcome. It keeps us on our toes.
Absolutely. Keeps those brains working, those labs humming.
Yeah.
We have to keep learning, keep asking questions, and keep pushing the boundaries of our knowledge. Because as we've seen today, even the smallest organisms can have a huge impact on human health.
Couldn't have said it better myself. Well, on that note, I think it's time to wrap up our deep dive into the world of Moraxella, catarrhalis, and Kingella kingae. It's been a real pleasure exploring these fascinating bacteria with you and our listeners.
To everyone listening, thank you for joining us on this microbial adventure. We hope you learned something new, maybe gained a new appreciation for these tiny but mighty creatures. Keep those minds curious, keep those labs running, and we'll catch you next time on The Deep Dive.